Hi, it's a great pleasure to start the second part of the morning session. And the next presenter is Professor Trevor Lamb, who will talk about visual neuroscience from light detection to macular degeneration. So Professor Lamb obtained a degree in electronic engineering at Melbourne University, and then he transferred to physiology before traveling to Cambridge in 1971 to undertake his PhD. In the early 90s, he and Edward Pugh at the University of Pennsylvania developed a mathematical description of the molecular reactions underlying the onset phase of the photoreceptors light response, which has provided important insights into the transduction mechanism. Most recently, he has investigated the evolutionary origin of the retina and photoreceptors. In 2006, they were joint recipients of the ARVO Proctor Medal. In 1993, Trevor was elected as a Fellow of the Royal Society, and in the following year he was promoted to a chair in Cambridge. In 2003, he returned to Australia as a Federation Fellow at the John Curtin School here at the ANU. Currently, he is Distinguished Professor at the ANU and he's Research Director of the ARC Center of Excellence in Vision Science, which links vision research at 4 universities, the ANU, Universities of Queensland, Sydney, and Western Australia. So it's a great pleasure to have Trevor here today.
Thank you very much, Julio, and thank you very much for the kind invitation to this very exciting meeting. The topics that I'm going to be covering are shown here, starting with basic research on the eye and moving through to tell you about other people's work that relates to translation. So I'll start telling you about the retina, then I'll spend time on photoreceptors, on their response to light, how they respond to light, and the cellular mechanisms, before moving on to considering some of the neural processing that goes on in the retina, and then getting to the more translational areas, retinal degenerations, what can possibly be done about them, and then on to prostheses and diagnostics in the eye for the visual system. Now, I'm just going to keep the arrow on. Okay, so I'll start with the eye and retina. Here's the eye as you all know it. The inner lining of the eye, shown yellow here, is the retina, covering most of the interior of the eye. And if we zoom in on the retina, we take a small section at the top there, zoom in, then schematically we see the following. Outermost are the photoreceptors, the rod and cone photoreceptors that actually respond to light, and they're going to be the bulk of what I'll be talking about today. In from them is a layer of processing cells, in particular retinal bipolar cells, but other cells as well, I'll come to later. And finally, the output cells, the projection neurons, which do more processing, the retinal ganglion cells, which send their axons out, so they run out here, out the optic nerve and up to the brain. What this actually looks like in the light microscope is shown here. So the rods and cones, the photoreceptors, their nuclei are shown by this nuclear layer. Another nuclear layer, inner nuclear layer for these processing cells, and finally a third nuclear layer for the ganglion cells. Now, it's interesting that light has to travel through the entire thickness of the retina before it hits the photoreceptors. If I just go back, light coming in here has to go through the whole thickness of the retina before it actually gets to the light-sensitive parts out here. And that has implications that we'll come to later. And this diagram just shows the positioning of the cone and rod photoreceptors, schematic sketches of those in human. So let's zoom in on the photoreceptors. These are rather nicely marked cone photoreceptors. This is from the monkey retina. The entire cone is marked through to its outer segment, the light-sensitive region here. And for the rods, which are much more numerous, only the outer segments, only the light-sensitive regions are marked in this section. So I'd like to zoom in on the light-sensitive region, the outer segment. So let's zoom into the outer segment, shown schematically here. So these outer segments sketched up here are shown schematically in a little more detail here. And they are comprised of membrane, evaginated membrane, folded out membrane here in the cones, which actually becomes sealed right over to form free-floating discs in the rods. And if we zoom in again, here's an EM showing these discs. There's a stack of something like 1,000 or so of these discs up the outer segment. You can think of it as flattened balloons, membranous balloons, surrounded by the plasma membrane coming down the two sides. And if we zoom in further from the outer segment, from the discs to the membrane, then we see that these membranes, again schematically, are chock-a-block full of protein. The great majority of it is the visual pigment, that's rhodopsin in rods, or the cone pigment in cones. In addition, there are other proteins important to transduction, but they're present in much lower quantities. And we can zoom in again on this photopigment molecule, rhodopsin in rods, and 10 years ago its crystal structure was determined and is shown here. It's a protein, 7-transmembrane segment protein, a member of the G protein coupled family of proteins, and it has a vitamin A moiety inside it, which actually absorbs the light. So light comes in, activates this 11-cis retinal and activates the cascade, gives you the electrical response to light. Now, that was photoreceptors. Let me move on to these electrical responses. I'll start by talking about what you can record in single cells. I'll come on to whole animal recordings of the electroretinogram after that, and in between I'll just briefly discuss molecular mechanisms. So here are the photoreceptors, a human rod and cone here, and here's just a schematic of a rod with this stack of discs down its outer segment. To record electrically, the approach that we take is to take one of these rods and to slip a glass pipette down over it. And that way you can record the electrical current that would normally flow. So an actual experiment is shown here. Here's an amphibian rod. This is actually a salamander rod drawn into a glass suction pipette, and this is ringer solution all around here. And what we do is simply connect a very sensitive current monitor between the inside and outside of that pipette, and we can record the current that flows in that cell. And what is found for the schematic here is that in the dark, there is actually a steady circulating current flowing. So there's a current flowing like that normally in the dark. You slip a pipette over and it flows through your recording system. So in the dark, there is this steady current flowing. Now what happens in light? You hit the cell with light. What happens is that that current is interrupted. You close down this current, so the response to light is a reduction in that circulating current. Now the responses that you measure with this technique, shown in the next slide. So here's the circulating current. This is the dark current level. In this cell, it was a salamander rod, it happened to be about 44 picoamps, 10 to the minus 12 amps, but this steady current flowing in darkness. You then hit the cell with light, flashes of light, running from either very dim up to very bright, and what you see is a graded suppression of the circulating current. So dim flash gives you a little suppression of that current, brighter flashes give more, and a bright flash completely shuts off that circulating current. So the take-home message for the electrical response is that light leads to a slow and graded reduction in the circulating current. Now, this was an amphibian rod. It was at 20 Celsius. Things are very slow. It's about a 1-second time to peak. Body temperature in our rods somewhere around 5 to 10 times faster, around about 100 milliseconds, rod, time to peak. Our cones are maybe another 5 times faster than that. So our photoreceptors are a lot faster than this. Now, what about the cellular and molecular mechanisms underlying that response? Well, first of all, this current that was flowing in the dark, what is the origin of that? That is actually carried by sodium ions flowing into the outer segment and potassium ions flowing out of the inner segment. Now, if that happened all the time, the cell would run down. That can't happen, so there has to be a pump. There's a pump that's pumping sodium out and potassium in. Sodium-potassium pump, or ATPase, it's continually consuming energy. And the photoreceptors are consuming massive amounts of energy. The photoreceptors have the highest energy consumption of any tissue per unit of mass in the body. They are massive drains on oxygen, and that turns out to be quite important in the viability of photoreceptors in the long term. What about the molecular basis of transduction? There is a G protein cascade involved. Rhodopsin is a G protein coupled receptor. Here's a sketch of an EM, sorry, not a sketch, an EM of the discs and the plasma membrane. And here's a schematic of the reactions underlying transduction. I'm not going to go into any detail here. It is complicated, but here's a sketch of a disc. So this is one of these membranes down here. So that's the disc membrane. It's chock-a-block full of rhodopsin. There is also a G protein present in lower concentration. There's an effector protein in the photoreceptor. It's a phosphodiesterase. And so these are reactions going on in the disc membrane. There is then a second messenger. Cyclic GMP is a second messenger that links the reactions in the disc membrane through to the plasma membrane, this membrane running down the outside that contains the ion channels, and the light response actually leads to closure of these ion channels brought about by changes in cyclic G concentration. Now, these are just the reactions for the onset of light. There's a whole lot more reactions involved in shutoff of the response, but a great deal is known about these reactions, and they have been modeled. And as Julia mentioned, Ed Pugh and I developed a quantitative model for the onset phase of the response to light by analyzing these reactions, ignoring shutoff, looking at the onset. And I won't go into the details, but just to say that it's possible to predict the form of the light response, at least the onset phase of it, from analysis of this model. So here were the responses I showed before. Because we can only consider the onset phase of the response, because in that model we didn't analyze the recovery phases, we can only look at the early phase of the response. If we expand those responses out, exactly these responses, just expand them out, those are the responses from that same cell. That's experiment. The predictions of our model are shown there. That's for the measured light intensities, and the model actually gives an incredibly good description of the onset phase of the response and gives us confidence in the model. Now, that was from single cells. What about the light response from the whole animal, and in particular from human? So here's the electroretinogram apparatus that we use for recording responses from human subjects. This is called a Gunsfeld. It's a sphere. This is Alison Cameron, who was a postdoc in the lab. There are some recording electrodes that connect up to her eye. There's a flash gun here for delivering flashes of light. There are some light-emitting diodes here for steady background lights. And there's a TV camera over here looking in at the eye. And what it sees is something like this, a dilated pupil, and this shows our recording electrode. This wobbly line here is our recording electrode. It happens to be called a DTL. It's a conductive fiber, silver-impregnated fiber, and that records electrically from the eye. Now this, as shown here, is a little bit artificial. We don't usually have it sitting above the eyelid. It's usually like this underneath the eyelid. So here's a DTL recording electrode coming in. It actually goes down under the eyelid, comes out, and we just tape it onto the nose here. And then it's connected via crocodile clip out to our amplifier. And our indifferent electrode is this skin electrode. So this is a conductive skin electrode that's connected to the negative side of the amplifier. So we're recording between this conductive electrode on the eye and just the skin, the head. So the way that works out, here's the eye. We have this conductive DTL electrode on the white of the eye, on the sclera here. We also have a skin electrode. What that skin electrode is doing is giving us a path to the outside of the eye. This DTL electrode is giving us a path to the interior of the eye, which is just salt solution, basically. And so we're able to measure the voltage across the retina using these two electrodes. And if I go back to the sketch of the retina here, we can now measure the voltage across this tissue. And as I mentioned, as far as the photoreceptors are concerned, there's a circulating current in darkness. That current flow gives a voltage drop. So we can measure that voltage drop from the photoreceptors here. And if we give flashes of light, we basically interrupt that current. We get a change in voltage, so we can measure that change in voltage from the photoreceptors in the intact eye. Now, also, there are other radial components here. They're going to be bipolar cells. Other cells will give responses. Sometimes it's difficult to separate them. But under ideal circumstances, we can separate these. So what do the responses look like? Well, here's a family of these electroretinogram responses. These are rod-isolated, so this is rod-only responses. The A wave, A is the initial response, the initial negative going response, and that basically represents the suppression of this circulating current here. And this is a series of responses. The black are experiment from very dim, progressively increasing intensity up to very bright. And for a line up above here, the top left, it's essentially photoreceptor activity, just the outer segment of the photoreceptor. At later times, we get the bipolar cells and other cells coming in. There's later activity coming in from other cells down here. But you can see again that the theory, this is the red traces, gives an extremely good fit to the onset phase of the response, and that, again, what gives us confidence in two things. Firstly, that the electroretinogram and its A wave is accurately reflecting the activity of the photoreceptors in the living human eye, and secondly, that the model gives a good fit to those responses. Now, that's in human. That's actually my left eye. You can do the same in just about any experimental animal, and these are comparable recordings from mouse. It is a very standard technique, and the theory fits. Let me move on now to some aspects of the circuitry of the retina and processing, and in particular the fact that the retina is divided into two separate pathways, on and off pathways. Now, that's the diagram I've shown you so far. It's a pretty simplified diagram of the retina. A more realistic schematic showing the cells that are there is shown here. It's very complicated. There are rods. In our eye, there are 3 spectral sensitivities of cones. I mentioned bipolar cells. There are about 10 different varieties of bipolar cells linking these through photoreceptors through to the ganglion cells. There are at least a dozen classes of retinal ganglion cells giving their output to the brain. And there are other cells I haven't told you about, horizontal cells making lateral interactions at the outer retina, and about at least 40 classes of amacrine cells mediating lateral and other interactions at the inner plexiform layer. So it's a complicated part of the brain. And this is a part of the brain. This is a part of the central nervous system. Now, as far as this wiring has been sketched diagrammatically here by Hans Wessler, And I will just concentrate on this pathway through the retina, from cone photoreceptors through bipolar cells to ganglion cells. It turns out there are two classes of retinal bipolar cell. There's an on division and there's an off division, and likewise two classes on and off divisions of most retinal ganglion cells. So each cone actually connects to two classes of bipolar cell. And here's a bit of a complicated diagram, but if I just hone in on the on pathway and the off pathway, here's a cone. It connects to an on bipolar cell and to an on ganglion cell, and that means that when light comes on, there's firing of this ganglion cell. Over here, this could be the same cone, also connects to another class, an off class of bipolar cells, to an off ganglion cell, and so when the light goes off, this cell fires. And so this is a fundamental division in the retina, and it's the start of the on-off division of the entire visual system. Just very briefly, one point about evolution of the retina, because there are some very exciting things about evolution of photoreceptors and evolution of the retina. I'm only going to mention one here, and that's in relation to this diagram, because it shows the pathway from cone photoreceptors through to ganglion cells. Now, this turns out to be the primordial organization in our retina. Cones were there first. The rods, which are, for us, the most numerous photoreceptors, there's a lot of evidence that they evolved later, and in particular that their pigment, rhodopsin, evolved after the cone pigment. And so the rod pathway is an addition. It's pretty clearly been added on after this pre-existing cone pathway, and so the rod signals share the cone pathway up to the brain. The simplest system for, say, twilight levels is direct gap junction coupling from the rod photoreceptors onto the cones and then just straight through the cone pathway. But down at starlight levels, there's a dedicated path from these rod photoreceptors through a rod bipolar cell, a complicated pathway involving something called an A2 amacrine cell, and then in onto the cone pathway. But basically, what's happening is that the rods have been added later. They've had to come up with a pathway that just piggybacks on top of a pre-existing neural pathway that sends signals through from the cones. But I won't go into details on that. And there's a lot of other evidence also that the rods evolved after the cones. Let me move on now to retinal degenerations, and I'll cover two classes of disease, retinitis pigmentosa and age-related macular degeneration, but I'll start off by talking about the normal retina. So normal retina, we have something like 120 million rods throughout our retina, about 6 million cones, the order of 5% cones. But the cones, as far as our functional vision is concerned, what's important is an area, a pit called the fovea, which is where we direct our gaze. Whenever we look around, we direct our eyes. What we're doing is directing our fovea to the point of interest. And the fovea contains about 100,000 cones. And these are crucial to our high-resolution vision. Here's a view of the normal retina, light micrograph of the normal retina, getting out a little bit peripheral here, compared with the sketch that I showed of photoreceptors, bipolar cells, ganglion cells. Here's the photoreceptor layer with their nuclei. Here's the inner nuclear layer, bipolar cell nuclei and others, ganglion cell nuclei down here. Now that's typically what you get out in the periphery. In this fovea, this pit, that's the region here, and it's a very specialized region. And what you find is that these superficial layers have been displaced from the fovea because light is coming in from this direction. it will be scattering light. They've been shifted to the side, and there's a specialized region here that has most of the superficial layers pushed aside and has incredibly slender cone photoreceptors, and it has no rods. And this is what you see if you look in here. You see a close-packed array, really dense array of just cones, and that's what gives us our high-resolution vision. And if we ever lose that area, if we lose those 100,000 cones, so if we lose basically 1 in 1,000 of our photoreceptors, but a particular 1 in 1,000, then we're functionally blind. Now, that's the normal retina. Photoreceptors basically exist on a knife edge. I mentioned before that they are incredibly hungry for oxygen, so hypoxia can be a serious issue. There's a massive blood supply out here in the choroid. There are some vessels on the surface here supplying the inner layer, but the photoreceptors are right in the middle, and they are really dependent on oxygen. Another issue to this living on a knife edge is that they are exposed to light and sunlight, incredibly intense. A lot of it, there's going to be UV getting through, and this is going to lead to the creation of free radicals, oxidizing agents, and so it's a nasty environment to be in. Low oxygen and free radicals. So they're living on a knife edge, and lots of things can go wrong and can lead to retinal degeneration. And what happens in retinal degenerations, as far as we're concerned, the important things basically are that you lose the photoreceptors. There are some other things can go. The retinal pigment epithelium can go, but functionally, the important problem is that the photoreceptors go. The two classes of disease that I mentioned, if this is occurring out in the periphery, it will typically be called retinitis pigmentosa. The photoreceptors out here have gone. The retinal pigment epithelium may be exposed, may be changed. That's out in the periphery. If there are changes in the macular region, these will be called macular degeneration, and in particular, age-related macular degeneration, possibly stemming from the fact that this area has been exposed to these conditions for maybe 7 decades. And in macular degeneration, if you lose these central cones, you are functionally blind. So, what does it look like? And if an ophthalmologist looks in here, looks in through the lens, looks at the back of the eye, this region here, the fundus, what does he or she see? And just for a landmark, the optic disc is a real landmark. That's where the optic nerve leaves. And so what you typically see, looking in at the normal eye, is something like this. This is the fundus. Characteristically over here, the optic disc. This is where the nerve fibers are leaving and the blood vessels are entering and leaving. Then a yellow spot. It doesn't look very yellow in this image, but macular lutea is the yellow spot. And the yellow is probably there to cut out ultraviolet and protect the most sensitive photoreceptors. And right in the center of that is the fovea centralis, so the central pit. And that's where this very high resolution area is. If you look at this fovea, you'll see that there are no vessels there. The vessels stop before they hit the fovea. It is an avascular region, and that's shown here. These images from Jan Provis showing the central avascular region. So the fovea doesn't have retinal blood vessels. It has to rely on the choroidal circulation, and this, again, may be a contributory factor to the degeneration of the macular region. So let me... not sure what I did there. But, okay, retinitis pigmentosa and AMD I'll kind of take together. Normal retina In retinitis pigmentosa, which is a horrible disease, an inherited disease, this is about the only region of the retina that is unaffected. This region with the fovea is not affected. There's all this black pigmentation out here in the peripheral region. The photoreceptors have been lost, the pigment is exposed, the pigment is coming through, and as a result, this is the only region that's working, so someone with that condition has tunnel vision. They can only see centrally. Retinitis pigmentosa is a heterogeneous set of inherited diseases. There are numerous known point mutations, dozens and dozens of them, that will lead to retinitis pigmentosa. Many of them are in rhodopsin, but there are many, many others as well. And mutations in rhodopsin don't always lead to retinitis pigmentosa. Converse to this, AMD, age-related macular degeneration, affects the macular foveal region. It's characterized by these spots called drusen, which are some of the earlier manifestations of this disease. It can be, this person will have lost their foveal region, and the debilitating thing is you lose the ability to read, to see faces, most of the things that we use for functional vision. This is a slowly progressing disease. Many of the elderly people in this room may have drusen and may be going on to develop this disease in later years. I include myself there. In contrast to this, something that can occur very rapidly, and this is what's called dry AMD, is wet AMD, which is neovascularization. There are blood vessels invade from the retinal pigment epithelium, and this is devastating, and this is irreversible. It's going to give the same kind of vision loss. So, looking at photoreceptor degeneration in AMD, here's the normal retina that I showed before. It's actually an elderly person. And here's AMD, similar-aged person. And basically what has happened is that the photoreceptors are all gone. There's no sign of a photoreceptor layer here. The inner nuclear layer is there. The ganglion cell layer is there. It's looking a bit mushed up. But the photoreceptors have gone. The fact that these layers are still present, the processing layers, is important, and as I come to bionic eye, it can be very important. Interestingly, it was shown about 5 years ago that there appears to be a role for the immune system. There was about 4 different groups simultaneously reported in PNAS and Nature, a SNP, in complement factor H, accounting for much of the susceptibility to AMD. And this complement factor H is involved in inhibiting the activation of this membrane attack complex. It's not known the role of how this system contributes to AMD, but I think it's very interesting, particularly in light of Professor Schwartz's lecture at Julio's meeting a week or so ago, the potential role of the immune system in this disease. Jan Provis, here at the ANU in RSB and in our Centre of Excellence, is a world expert in AMD. And I'll just very briefly mention some of the work that Jan is doing. She's interested in the genes that are involved in the development of the macula and potentially in its degeneration. And they do gene array work. And they take different regions of the retina, developing retina, fetal retina, and they look at the genes and analyze what they see. And basically, they've been looking for genes which are expressed in the developing macular region, and after a lot of analysis, they've come out with a number of genes that are regulated. In particular, regulators of angiogenesis, that is of blood vessel development, and of guidance factors for axons and vessels. And they've found a number of potential leads here that are involved in development of the macula, maybe involved in degeneration, and two particular ones that Jan's group are following up at the moment are FA6 and PEDF. So there is potential here for translational work. Also in our centre, Keely Bumstead O'Brien is looking at development of the macular region, not in human, but in other animals that have fovea, notably the pigeon and the seahorse, because there are seahorse species that do have foveas and those that don't. And there's a lot of potential for seeing what it is that leads to a fovea, how you get a fovea, and what may be involved in keeping it there. Other work in our centre involves therapies, potential therapies to protect the retina. And Christina Volta is working on a number of potential therapies here that fall into three main areas. Light management, and that is basically seeing if by keeping light levels down you can prevent retinal degenerations, and for some of their models of RP they can indeed do that. Second avenue is using near-infrared light, which has been shown to aid healing. Whether that can prevent or even help to cure retinal degenerations, and it's looking very promising. And certain antioxidants, in particular saffron, again appears to have great potential for preventing retinal degeneration. Let me move on just now to prosthesis, so bionic eye. And this is work of Michael Ibbotson, again in our centre. And here the aim is not to prevent degeneration, but to do something after degeneration. And the photoreceptors are dead, but the inner retinal layers, the bipolar cells and the ganglion cells, appear to be alive. And so the basic idea is to put a stimulating array over the degenerate area and electrically stimulate and send signals up to the brain using these residual pathways. So Mike is a member of Bionic Vision Australia that recently got $42 million from the government, and this is a system that's basically been developed by Greg Swanning, and this is what they're planning to put into the human eye in the next couple of years. Here's their current array. I think it's 96 stimulating electrodes. Here's the realization. Those electrodes are out here. And that will be put in a person's retina. It'll be slipped in behind the retina here to stimulate the cells down here. Now, one of the problems with all these bionic eye approaches is you don't really necessarily know what you're stimulating, because there's an on and an off division of the visual system. And what Mike's basic research is doing is looking at how these stimulating arrays are going to be exciting the on division of the visual system and the off. And one aim will be to get optimal protocols for selectively stimulating the on pathway versus selectively stimulating the off pathway. The next step that they're going on to, the next generation, is going to have 1,000 electrodes, 32 by 32 array. It might give images something like this. It's going to be using a diamond substrate with these little pins sticking through and providing excitation to the retina. Visual diagnostics. Ted Mattis, again in our centre in RSB, has developed an instrument called the True Field Analyzer, which does what's called multifocal perimetry. And the instrument it works as follows. The observer looks in here, And then there are some light-emitting diodes that are illuminating the eyes with infrared light. The image from the pupil is coming back to a pair of TV cameras and is being analyzed. And then there are some stimulating screens inside this device so that stimuli are projected onto the eye, and the computer monitors the response of the pupils to these stimuli. And if it works, this is the kind of stimuli that are used. These are pseudo-random flickering stimuli, so the two eyes are being stimulated independently of each other, and then the computer analyzes the pupil constrictions to work out which parts of the visual field are functional and which parts are not, without any user intervention. These are kind of patterns that can be used. The device has FDA approval. It's getting close to being marketed. I think it is, a prototype is on the market, and it's actually very promising for the early diagnosis of glaucoma, particularly normotensive glaucoma. And you can look it up there. Okay, so just by way of summary, photoreceptors, we understand a lot about how they work. We can model their responses, we can record their responses at the single cell level, we can record their responses non-invasively in the living human subject. And in retinal degenerations, it is the photoreceptors that go wrong. That's functionally what goes wrong. And for translation, we really need to look at ways of preventing photoreceptor death, or preventing death or regrowth of those that do survive. As a perhaps temporary approach over the next decade or two, the bionic eye as a prosthesis may be able to help, but the long-term aim must surely be to prevent these degenerations. So just by way of thanks, the people that were involved in my group here in John Curtin School, and the members of our Centre of Excellence currently located in RSB, but all these people will be moving in, or scheduled to move into Stage 3 of the John Curtin School in 2012. Thank you very much.
I'd like to thank Trevor for this very inspiring presentation and open the floor for questions. I have a very simple question in terms of it's almost like, almost ashamed to ask, but in terms of people living longer and longer and the expectation of life being like 80 years now, that it is not what we were evolved to do, what's the actual kind of harmful effects of direct sunlight, like people who live in very bright environments without shades through 5, 6, 7, 8, 9 decades? How bad can sun be for that region that you showed?
This is a point of considerable debate. There's a lot of evidence to suggest that a lifetime exposure to sun is extremely dangerous. Jonathan Stone, a member of our centre who works on exactly this, I guess in his 7th decade, has religiously been wearing sunglasses for about the last 10 years. So somebody who works on the topic certainly believes it's important. And there is quite a powerful case for thinking that we are basically outliving our eyeballs as we live longer and longer. Vaughan? No.
The understanding of why the photoreceptors are in the back of the eye. I mean, obviously, is it an evolutionary advantage, or
yeah. Okay, it's the way our eye evolved, and it turns out the path that our eye went down, it was the only way that things could evolve, and it's basically for getting the retina in contact with the retinal pigment epithelium. And if you look at the development of the eye in utero, the way the optic vesicle sprouts out and then the eye vesicle invaginates, this is the only way of getting the photoreceptors in contact with the retinal pigment epithelium. And they need that because the retinal pigment epithelium is what regenerates 11-cis retinal. It does the vitamin A cycle, and they have to be able to get their pigment regenerated. It's a long and complicated story. I could go into details. I'd be happy to at length. But it's basically the path that the vertebrate eye took in evolution, very different from the path that invertebrate eyes took. But I believe it's basically to put the outer segments in apposition with the retinal pigment epithelium for regeneration of visual pigment.
Any other questions?
Trevor, with respect to the retinal prosthesis, are there sufficient biophysical differences in the on-off pathway that will allow focal stimulation and convolution over the eye?
That's what Mike and other people have got to look at. But I think it's possible from the timing of the stimulus and maybe how you pair stimuli, or that I think they're going to have to look for ways of differentially stimulating the on and off pathway. And that's basically what one of the things that Mike's looking at the moment, because they are implanting those stimulating arrays in cat's eyes, recording from cat cortex. And they've already been able to show that signals get up to the cat cortex. They now have a lot more work to do in basically optimizing the stimulus. Because what you don't want to do is stimulate so your brain gets signals saying on and off at the same time. Now, whether it's going to be possible, I don't know, but that's one of the things they really have to work on.